Hello and welcome dear viewers to the informative video on bioavailability and bioequivalence studies involving the study of food effect. As we know that bioequivalence is the key requirement for product approval, whether it may be generic formulation or a new formulation or novel formulation, or you can say it as NDA, that is new drug application, or you can say it as an innovator formulation or reference formulation. So whenever the generic product is required to be approved, that time bioavailability studies and bioequivalence studies are important. Mainly for generic products, bioequivalence studies are done with the reference product as a standard to compare the generic formulation. Many of the times the bioavailability and bioequivalence studies are performed in fasted state and in fed state. So these two states are studied to know the impact of food on bioavailability and the bioequivalence. So let's start with the video and see how the studies are performed, why these studies are required, and what are the requirements. So why food effect studies are performed? See, for the oral formulations, food in the stomach and intestine may have effect on the drug absorption. So food may affect the bioavailability by impacting the rate and extent of the drug absorption. One thing is required to remember here, that food may impact rate and the extent. Both these things are impacted by the presence of food. So rate of absorption means Cmax and extent of absorption means AUC. And how the presence of food has impact onto the drug bioavailability. So presence of food may delay the gastric emptying. There is difference in the gastric emptying time in fasting and fed state. Then the food may interact physically or chemically with the drug. Some drugs may form complex with the food or food constituents, or these may get interacted physically, altering the gastrointestinal pH. So this is another important reason that the fasting GI pH will be different compared to the fed GI pH. And here I am referring GI pH means consider it as pH of the stomach and the pH of intestine. So this pH will be different in fasted and fed state, and that's why this change or difference in the pH may have impact on to the drug bioavailability. And also consider, if your drug is having pH dependent solubility or the formulation is designed to have pH dependent release profile or dissolution profile, then the change in pH because of food may have great impact. Then stimulating the bile flow. See, the bile contains the constituents which may increase the solubility, like bile salts are there. So these bile salts may increase the solubility of some of the APIs or drug substances, and thereby they may have changed or may have impact onto the drug solubility and absorption. Then altering the luminal drug metabolism. So if there is a luminal gut metabolism or luminal drug metabolism, that time these fasted and fed conditions will have different drug metabolism, and thereby the bioavailability will be different in fasted and fed state. That's why the bioavailability and bioequivalence may be different in fasted state and the fed state. So because of these things, food effect bioequivalence and food effect bioavailability studies are performed. See here, one thing is important to understand and note that generally the food effect bioavailability studies are performed for the new formulations, and new formulations made of new chemical entities or new drugs or novel molecules intended to be given by the oral route. And food effect bioequivalence studies are performed mainly onto the generic formulation, and for some of the formulations with change in composition, change in manufacturing process for the products for which already the fasted and fed condition studies are done in clinical studies. Then food effects becomes complex because of which type of reasons. So it is not very easy to understand the impact of food. We cannot predict what will happen because of the presence of food. So food effect is complex because of volume, differences in volume, temperature, nutrients, calories of meal, which affect the drug product transit time, luminal dissolution, that is dissolution of the formulation inside the GI tract, drug permeability, and the systemic availability. Then the high calorie and high fat meal has much more effect onto the bioavailability and the bioequivalence. See, one thing is here to understand that high fat meal. So US FDA, EMA, and many of the regulatory authorities and regulatory bodies have given the clear information for food, food constituents, and how much calorie food should be used, how much fat content should be there for performing the food effect bioavailability studies and food effect bioequivalence studies. Now, consider a high fat meal or high fat food, and you take the molecule or drug or formulation containing the drug which has higher solubility in the oil phase. Then what will happen? As a general thing, the molecule will get partitioned into the fat part or oily part of the food, and then it may get absorbed at a higher rate or at a lower rate, or its rate may be changed compared to the fasting state. That's why to have the clear understanding and to have clear idea about the behavior of the formulation and the drug molecule, high calorie and high fat meal is proposed for performing the food effect studies. The food may increase or decrease the rate and extent of absorption and may have significant clinical effect. Now, what is the meaning of this significant clinical effect? So, if the rate and extent of absorption is changing because of the food impact or because of the food effect, then sometimes there will not be any impact at clinical front, or sometimes the due to the increase or decrease in the rate and extent of absorption, the therapeutic outcome may vary. Sometimes due to higher absorption in presence of the food or in presence of the food with high fat content, toxicity may be there. Sometimes if the rate of absorption get doubled, then think that your Cmax and Tmax will vary, and because of that variation, the safety and efficacy may get compromised. So, food effect studies are very important. The use of high calorie and high fat meals is suggested for food effect study, and this is the reason, as I have explained, it might have significant clinical effect or it may compromise the safety and efficacy. The product label should have the instructions for the administration based on the food effect. Now, if high fat meal increases the Cmax and Tmax of the formulation or drug molecule, then it should be written onto the product label that avoid high fat meal consumption when you are taking this medicine, or whenever you are taking this medicine, avoid the high fat meal. So these type of instructions are given onto the formulations. Now you might have a question that if the food effect is there due to high fat meal, how to perform the food effect studies? Because as I have mentioned earlier, food effect studies are performed with high calorie or high fat. So dear viewers, please remember that whenever there is a high fat meal effect onto the formulation, safety and efficacy, that time these formulations are tested with the low fat content meal in the food effect study. So this is very important. Whenever you come across a situation where your molecule or your RLD suggests that avoid high fat meal for administration or for dosing, that time you have to perform food effect studies with low fat meal. So please remember these things. Then food effect bioavailability study to be done early in the development phase to derive the dosing conditions, that is fasting or fed conditions for administration. Because if the food effect study is not done in early phase, then how the sponsor will know that what state is to be used for doing the other clinical studies and the bioavailability studies. So that food effect study is required to be completed and performed in early in the development. To understand the clinical significance of food effect based on the food effect on drug absorption and bioavailability. So Cmax and AUC differences are required to be studied with food and without food. Food effect studies are required on all modified release formulations. Whenever there are modified release formulations, that time always food effect studies are performed. Generic formulation and the formulations with variations are required to show bioequivalence in fasting and fed. So this is dependent onto the formulation. Always this will not be the case, but for all modified release formulations, food effect study is required. Here BCS class and the formulation design considerations for food effect study. Consider an example of BCS class 1 drug formulated into IR formulation with rapid dissolution. For example, rapid dissolution or very rapid dissolution. Consider more than 85% drug get dissolved within 15 minutes. For both the formulations like RLD and the test, that time this type of formulation will be less sensitive to food effect as the BCS1 drug shows pH and site independent absorption, and the differences in the dissolution will not have much impact onto the bioavailability with food and without food. If there is a high first pass effect, extensive absorption, complexation or instability of the drug substance in the GI tract, then food effect may be seen. So if your molecule is unstable in GI tract, that time you may see the effect of food. The similar food effect can be expected for the test and reference product. See here, if you are working on generic formulation, so that time your formulation design will be same and the API will be same. That's why you can expect the similar behavior of the test and reference formulation in the fed and fasted state. Then BCS3, BCS2 drugs, BCS4 drugs formulated into higher formulations may have food effects because BCS2 and 4 are insoluble type of molecules and BCS3 is having the permeability limitations. That's why it is not like the BCS class 1 drugs. These BCS class 2, 3 and 4 molecules formulated into higher formulations may show food effect. And MR formulations. All the MR formulations may have food effect. Higher formulations are generally required to show bioequivalence in fasting state as per the Europe requirement. But for the same formulations, for US, both fasting and fed conditions are required. And this is very important point to understand that for Europe, generally fasting state is required and for US, both fasting and the fed conditions are required. New formulations and the new drug like NCs or NMEs, food effect study recommended in early development to derive administration and food intake instruction on the label. Then generic IR formulations, fasting and fed study recommended, but only fasting study required if the IR formulation with BCS class 1 drug and dissolution is rapid for both test and RLD. Then RLD label states that the product should be taken only on the empty stomach. So this means that RLD is suggesting to take the formulation on empty stomach. That time generally bioequivalence study is not done on the fed state. Then RLD label does not make any statement about the effect of food on the absorption and administration. So in this case also you can go directly for the fasting state study. And for all MR formulations, both the studies are required. Then all new generic formulations with MR design are required to show bioequivalence in both the states, that is fasting and fed, to have the understanding of food effect. See, some of the formulations with MR design may undergo dose dumping due to the food effect. That's why MR formulations are required to show bioequivalence in fasting and fed state. Then study design for the food effect. So generally the crossover study design is used. Those study designs are called as randomized, balanced, single dose, two treatment, that means fed and fasting state, 2 period, 2 sequence crossover design. And subjects can be selected from the healthy volunteers or healthy population, and patients are included if safety is the concern. See, other requirements for the food effect bioequivalence and bioavailability study are similar to the generalized bioequivalence requirement. Like you can take higher strength for food effect study, or you can select the strength based on to the pharmacokinetic linearity, safety profile of the molecule, and the sensitivity for the analytical method. Then as per the recommendation of the reference product, you can go for the fasting or fed state. Then the Fasting state is considered as a reference state to compare the fed state. So if you are comparing the fed state, then fasting state results of the bioequivalence study will become as a reference and can be used to compare the fed state. So always see the product labeling, what are the administration and dosing requirements and the instructions of the reference formulation, and based on that try to understand the effect of foods, different food, different population, and also read the patient information leaflet of reference product thoroughly. BA and BE related questions are always asked in the interviews. So for interview preparation and learning, stay, stay tuned to Pharma Learning in Depth channel and keep on watching the videos. Thanks for being with me, and we will meet you soon with such kind of important and informative videos. Thank you.